All right. Hello, everyone. I wish you all a very happy new year. And here we are with the brand new year and with our speaker, Dr. Paul. This is a IASNM MISI webinar, and I welcome you all on the webinar. Just a few housekeeping points before I introduce my distinguished speaker. So it's going to be a 50-minute talk followed by a Q&A session. And if you have any questions, please feel free to put in the chat box, and you are more than welcome to ask those questions live at the end of the session. During the session, kindly please mute your devices and phones to avoid any interruption. Now it's my absolute pleasure to introduce Dr. Ramsey PAULMURUGAN, who is a professor of radiology at Stanford University School of Medicine, and he's director of the Cellular Pathway Imaging Lab. He's a member of the Molecular Imaging Program at Stanford, the Canary Center for Cancer Early Detection, Stanford Cancer Institute, BioX, and the WuSci Neuroscience Institute. Dr. Paul received his MSc in Biomedical Genetics and Ph.D. in Microbiology from University of Madras, India. After research positions in India and at UCLA, he joined Stanford, where he has been a faculty member since 2009. He has over 25 years of experience in molecular and in vivo imaging, including bioluminescence, fluorescence, PET, SPECT, ultrasound, MRI, CT across small and large animal models. He has published more than 250 peer-reviewed articles, authored 27 book chapters, and filed 9 patents, and has an H index of 60. His research pioneered split reporter protein complementation system for imaging protein-to-protein interactions and cellular signaling in living subjects. His current work focuses on molecular imaging of cancer biology, estrogen receptor signaling, chemoresistant pathways, and neurodegenerative diseases. Dr. Paul also leads translational research in cancer therapy, including microRNA-based reprogramming of drug-resistant cancers and ultrasound microbubble-mediated targeted delivery strategies. He's a recipient of multiple scientific honors, including Distinguished Investigator Award in 2018 from the Academy of Radiology and Biomedical Imaging Research and RSNM. And without further ado, Dr. Paul.
Thank you, Saima, for a nice introduction, and good morning and good evening to everyone around the world, and also Happy New Year to everyone. Thank you for, first of all, I would like to thank for the invitation to give in this particular platform. So today I'm going to briefly discuss about one of our recent work on how we can use like a kind of cellular vesicles as a multiple platform in drug delivery and RNA delivery as well as for immunotherapy. So I will give like a kind of brief background on what is this vesicles and how we can apply in multiple areas of different disease biology. So before I go into the talk, I just want to give a brief background on my lab. Actually, my lab is called Cellular Pathway Imaging Laboratory, and we work on multiple areas related to biology. We develop new therapeutics, so we can use them for like a kind of various treatment applications, not as just limited to cancer. We also use them for various infectious diseases. And we also work on like a kind of drug resistant mechanism in cancer, so that how we can overcome the drug resistance to improve low dose chemotherapy and radiotherapy. And we also work on the basic science, where we try to understand actually how the chromatin remodeling is happening in cancer, and they are contributing for drug resistance and treatment response. And we also work on developing new gene and cell therapy mechanisms, and we use like ultrasound for imaging as well as diagnostic imaging as well as targeted drug delivery in cancer treatment. And we also developing some sort of p53 refolding drugs, so that most of the cancers we know that actually p53 is mutated. This is considered a tumor suppressor gene. So when you kind of reactivate this particular protein, cancer cell respond to low dose chemotherapy. One or other way, most of our approach are trying to like a kind of change the existing treatment approaches so that make them more responsive without like a kind of developing new agents. So today my talk is like a kind of how the natural cell-derived vesicles can be modified in such a way that actually we can use them as a like a drug delivery agent, or you can also use them as a like a kind of a immunotherapeutic agents in cancer therapy. So here you are seeing like a cross section of a mammalian cell. So you see that actually there are various different cellular organelles which are sitting inside. And in addition, actually, they also modify according to the environment. So when the cells actually, it is not like a kind of similar to what we live, they are also experiencing various type of intracellular stresses as well as extracellular environmental mediated stresses, so that cells are like a kind of basic units. They are reflecting the whole body, how they are undergoing during various process, so that they tend to produce or excrete most of the toxic chemicals, as well as they also create new vesicles, so to communicate with neighboring cells or the cells which are located distant part of the body, so that during that process they develop a lot of different the cell derived vesicles. See, you can see here actually they are like a kind of forming from the cell membrane. They can form sometime towards outward kind of blobbing, or they can go invagination and they can form the vesicles, so that they are like a kind of organized process to kind of survive within this stressed environment, or intentionally they produce these vesicles to communicate with other cells. So here you can see that how these vesicles are produced by cells as a kind of platform to communicate with neighboring cells. So you can see here actually there are several different type of secretory vesicles. They also kind of release proteins. They can produce this kind of vesicles. These vesicles can also kind of form through the outward butting actually, so that it is filled with intracellular components. In some cases, actually, they are forming inside the cells. They undergo through lysosomal pathway, so that they are targeted loading of several different macromolecules within the cells, and they are released to communicate or deliver to the other cells. So that cells are like a kind of constantly changing this process, so that those vesicles are not like a kind of random vesicles. They are holding a lot of different biomarkers. Currently, they are used for different diagnostic applications. Even in the cancer, they can be like a liquid biopsy. You can see these cancer cell-derived vesicles in the circulation, so that you can just like predict after treatment whether they are forming, what type of vesicles they are forming, and what kind of biomarkers they are carrying. Accordingly, you can decide that your treatment is working or not. So the major types of vesicles normally cells produce are microvesicles and apoptotic bodies and exomeres. So these are like a kind of three major vesicles with different sizes. Based on their biogenesis pathway, actually they tend to carry different type of proteins on the surface. So, and also the loaded contents also, which include various RNAs and microRNAs. In some cases, if it is apoptotic bodies, they carry a lot of different DNA. So accordingly, you can use these cell-derived vesicles for different applications. So here I am going to talk about the three different applications. So one is like a kind of how we can use like a microvesicles and exosomes to load microRNAs for cancer therapy. In other cases, how we can use apoptotic bodies to load antibiotics in treating intracellular pathogens like especially bacteria. And also I want to show that actually these vesicles are displaying various different cancer cell specific surface antigens, how they can be used as like a kind of tumor specific vaccine for cancer immunotherapy. So as I said, actually, they carry a lot of different cell surface proteins, and all these cell surface proteins are depends on the source cells actually. They tend to show that what type of cells these vesicles are enriched. Accordingly, they will carry different type of biomarkers inside the vesicles as well as on the surface of the vesicles. Suppose if you want to use like a kind of cancer specific immunomodulations, you can isolate the vesicles from the same cancer cells so that they tend to express a lot of cancer specific antigens, and those antigens can be used to modulate immune system so that you can activate the immune system. Naturally, immune evasion is one of the mechanism by which cancer cells are surviving, but by adopting or putting like a kind of lot of targeted antigens, they tend to activate anti-tumor response in vivo. So you can see here the characteristics features of all these three different major vesicles. So you can clearly see that depends on the type of vesicles, their sizes are different, and also the cell surface by which they carry different proteins are different, and inside what they carry also different, and also that actually what are the pathway by which they are generated. So the extracellular vesicles biology goes all the way back to 18th century. But even though actually people thought that there's a kind of randomly released by the cells, but recently after the identification of exosomes in 1981, actually the pathway or the mechanisms or the applications related to the use of extracellular vesicles are like a kind of exploding. Now we use these particular vesicles since we have a lot of different technologies, how we can reconstruct them to the required function and load drug or load microRNA. So the application is like a kind of growing very rapidly. So as I said, actually, because these particular cells are carrying different biomarkers and different intracellular biomarkers like RNA, microRNA, as well as the cell surface proteins, they are specific to the source cells in from which they are released, so that they are serving as like a kind of diagnostic markers in cancer. And also you can like a kind of see that actually by identifying these biomarkers, we can say that whether they are coming from senescent cells, or they are coming from neurodegenerative cells, or they are coming from nerve cells or other pathological cells. So accordingly, we can use this the diagnostic biomarkers of these vesicles using like a kind of isolating from the blood or from the urine. Any of the liquid biopsy can serve as a biomarker. So the first application what I am going to discuss here today is how we can use these particular extracellular vesicles in like a kind of loading microRNA and or application in cancer therapy. So as I said, actually microRNAs are like a kind of small non-coding RNAs of like 18 to 21 nucleotides length. So it is easy to synthesize. You can make like a kind of most of these RNA or DNA, they are coming from the 4 nucleotides. So that it is easy to make, and there is no like a kind of huge toxicity or anything related to these microRNAs. So anything you adopt these small RNA-based therapeutics, they can be easily synthesized, and you don't need to go through like a huge kind of regulatory process, since they are like a synthesized from just 4 nucleotides. They are mimicking similar to exact nucleotides what we have inside the cells. So in this particular case, actually, we use microRNAs. The workflow normally we identify what are the microRNAs. They are dysregulated in cancer. They are selective to each cancer, so that we look for what microRNA they are regulated. If they are upregulated, we call them an oncogenic microRNAs. If they are downregulated, we call them tumor suppressor microRNAs. Accordingly, we use either supplement microRNA mimics to improve the cancer response to treatment, or we block the endogenous microRNAs which are upregulated in cancer. So by using antisense microRNA, that is called endogamase. So it is easy to identify microRNAs, so we can use those microRNAs for cancer therapy. So the first application where what we did is actually we exploit the pathway which is regulated in cancer cells. Cancer cells tend to express high level of cytokine, actually it is called STF1 alpha. This particular cytokine or ligand is specific to the receptor called CXCR4. So as I said, actually in this the extracellular vesicles released from the cells are depends on the nature of the cells or the protein displayed on the surface of the cells. So source cells are important for isolating these cell vesicles. So you have the flexibility. You can also modify the source cells in such a way that you can introduce new protein to be displayed on the cell surface. So accordingly, those vesicles released from those engineered cells can also carry the particular receptor which we introduce or we engineer them. So in this case, actually, we selected neural stem cells for treating GBM. So neural stem cells are like a kind of particular type of cells which are present in the nervous system, and they normally tend to go and attack different pathological region of the brain. So that in cancer also, you can see more and more neural stem cells accumulate within the cancer. So they have the natural property or tropism towards the pathological region, so that we engineered the neural stem cell by using CXCR4. Naturally, these cells express at a lower level of this particular protein, but in this particular case, to enrich their attacking or their interaction with the cancer cells, which are secreting these ligands, we overexpressed these cells. And we isolated these particular EVs coming from these engineered cells. And we have developed a technology called microfluidic based reconstruction process. In this particular process, we can like a kind of mix microRNAs or truck along with these EVs. And if you inject through this microfluidic process, they tend to reconstruct and load the RNA or truck within these vesicles. So by using this strategy, you are able to homogeneously kind of make uniform size vesicles of particular size between 50 to 100 nanometers. They are very useful for application in various delivery, either intravenous or intranasal or intraperitoneal, whatever be the application, you can inject or you can deliver these vesicles. So this particular microfluidic process, in principle, how they work, actually, you can kind of mix both vesicles as well as a drug or the microRNA with the input. And it has like a kind of very narrow channel kind of lined with a diamond, and it's a T junction. In this particular diamond narrow nanometer channel, when these microvesicles along with the microRNA goes through, this will be completely kind of disintegrated into pieces, and they are reconstructed during this process. During this reconstruction process, they tend to load the truck or microRNA which we mixed with these vesicles. So by using this process, it is like a kind of very rapidly you can make different type of truck loaded vesicles. So we use this strategy and kind of loaded microRNA within these vesicles. So in this particular slide, you can see here the vesicles isolated from these neural stem cells. You can see there is a wide range of vesicles from micrometer to nanometer size is present. It's a very heterogeneous. This is naturally isolated one. So after we reconstruct by using this microfluidic system, you can see here actually they are very, very uniform sized particles of something like 50 to 100 nanometers. So you can see here, this is another engineered vesicles. This is controlled vesicles and this is engineered one. But after reconstruction, you see very homogeneous vesicles. So to confirm that actually when you overexpress the CXCR4 receptors, whether they have after reconstruction, whether they still displays this kind of CXCR4 receptor on the surface, or they are like a kind of embedded within the vesicles. So that to confirm that actually we used a particular technique called immunostaining by using immunocold antibodies. So that this is like a kind of anti-CXCR4 antibody conjugated with a 10 nanometer gold nanoparticle. So that we stained this vesicle by using those antibodies, and we tested by using TEM. You can see here where is your the 10 nanometer gold nanoparticles. This is the vesicles. You can clearly see that compared to control, where you also see some of these particles, the one which we overexpressed, we see high number of this particle displayed on the surface. This clearly supported that actually after reconstruction process, the receptor which we express on those cell vesicles are still displayed on the surface, so that they can very useful for targeted delivery into the tumor. So here the some more like a kind of reconstructed vesicles for the understanding. So that different time point, number of time actually repeated so that we are able to see very uniform size vesicles. The average size is around 57 nanometer, but they are like a kind of 50 to 100 nanometers. So this is very optimal for drug delivery to different tissues. So when we work with any kind of therapeutic kind of monitoring process, we also introduce a lot of different imaging applications so that you will able to monitor all the process by using in vivo imaging. So to facilitate this process, what we did is we also labeled the vesicles by using ICG. So that ICG is one of like a kind of highly near infrared fluorescent dye. So we will be able to kind of image the animal after we delivered by using this vesicle by using optical imaging. So we also loaded the microRNA. Those microRNAs are conjugated with the sci-fi dye so that we'll able to see that whether those vesicles are carrying this microRNA. So that these are all the routine process which we use to kind of control every step involved in the reconstruction process. And you can clearly see that after we label the EVs by using ICG, you can see here all the proteins are labeled. And similarly, after we loaded the micron sci-fi microRNA, we isolated from the reconstructed vesicles. You can see that sci-fi microRNAs are present. Since I said actually microRNAs are like a kind of 18 to 22 bases, they are very short, 7 kilobases, so that you can see here. And we also imaged after before and after reconstruction for ICG as well as for the the sci-fi microRNA and ICG on the cell membrane. We confirmed by FACS analysis also. All this process clearly supporting that the microRNAs are loaded within the vesicles and vesicles are so labeled with ICG. And we tested the concentration of particles so that we'll able to control number of particles we inject by using NT analysis. And the DLS is to just measure the charge of those particles. Most of the time, any cell-derived vesicles are in the negatively charged. But when you load microRNA, nucleic acids are negatively charged and they tend to increase. So that it is clearly showing that actually the vesicles are in the negatively charged as well as the microRNAs are increasing the charge. So they are loaded or they are integrated into the vesicles. So first we tested how effective these vesicles in cancer cells. So we used like a kind of GBM in 3D culture so that this will facilitate, you can see in different layers of the cells how effective these vesicles are like a kind of targeting different GBM cells within the 3D culture. So the first one is like a kind of live cell stained with a nuclear stain. And this is like a before and after delivery by using ICG labeled EVs and the sci-fi microRNAs. This is an overlaid image. And we have used like a one with like a without engineering with engineering. That means this will have low expression of CXCR4 and this one with high expression of CXCR4. You can compare between these two. The one with the CXCR4 over expression, which is showing very strong and penetration into the deeper layer of the 3D culture. And you can also see in both the windows, this is the microRNA delivery. And the fusion is also showing that clearly it is targeting different layers of the cells. After we did in 3D culture, we also tested actually how they are delivered in the 2D culture. And we also looked into whether they are able to like induce any cell death. And normally when we use any GBM cells for this kind of study, we pre-label them by using luciferase or GFP protein so that you'll able to monitor the cells in culture as well as cells within the animals by using optical imaging. And you can clearly see here when you have more and more of CXCR4 receptor and they show like a kind of more TMC response. TMZ is the temozolomide is one of the commonly used clinical drug for GBM treatment. And you can see a lot of fragmented cells, they are dying because of TMC treatment. And that is sensitized when the presence of microRNAs. And here we see that in the location of this delivered microRNA inside the nucleus. Most of these microRNAs are located in the cytoplasm. So the advantage in microRNA mediated therapeutics actually, they don't need to go into the nucleus for any regulatory mechanism because microRNAs are regulating the mRNA expressed inside the cells. Most of these mRNAs are in the cytoplasm, so that it is easy to deliver and achieve high efficiency in treatment approach. So after we test them in vitro, we also went on to check actually whether they have equal efficiency in vivo. We used GBM cells implanted, orthotopically implanted intracranially in the brain, so that these cells are expressing luciferase, and you can easily image the tumor growth by luciferase imaging. And our vesicles, which we reconstructed, also having the fluorescent ICG dye as well as a sci-fi microRNA, so that you can also image them by using fluorescence imaging. So we have three different conditions. One is like without any treatment. Another one is with EV, control EV plus TMC, and EV with the TMC, microRNA alone, then combination. So total we have 6 conditions. And if you just look into the last one, where we have the complete treatment approach, and the tumor response showed much higher when compared to other conditions, because we had initially same number of animals. Wherever you see empty spaces, that means those animals kind of died early on. So we saw that actually when we had the complete treatment approach, where microRNA plus TMC delivered by CXCR4 receptor, there is a significant improvement in the survival rate. And we normally test them actually whether they reach the tumor inside the kind of in vivo conditions. We did bioluminescence imaging as well as fluorescence imaging ex vivo. And we found that most of these microRNA delivered by the EVs are co-registered along with the luciferase that indicates the tumor location. So as I said, actually, because anything we delivered via intranasal route, they tend to go some into the lungs, because it all depends on the breathing rate of the animal. So to address actually how much time these intranasally delivered vesicles are reaching the tumor site, we did another independent experiment where we kind of sacrificed the animal at a different time point after intranasal delivery of a single dose of EVs, and tested them for like a kind of the tumor location as well as the microRNA delivered by the EVs. So this is the one actually after 30 minutes after a single dose of EV delivery. So this is the luminescence imaging, which is indicating the tumor location. And this is the nose side actually. This is the after 30 minutes after intranasal delivery, you can see that most of these EVs are located in the front side of the nose. So still they are sticking to the mucosal layer. And there is a slight kind of, they go into the circulation, they accumulate in the lungs as well as the spleen. So if you look in 2 hours post, like a kind of, they are coming from independent animals, so don't relate with the previous animal. So this is another set of animal where 2 hours post, like a kind of delivery single dose, and this is the tumor location, and you can see here that EV started moving into 2 different directions. So they are moving directly to the tumor, and they are also going to the back side of that I will come to the details later. So we also did the same 12 hours post injection. Now you can see the tumor location is the same, but the EV enrichment is going more towards the back side as well as the front side. This is the tumor location. And when we did the 24 hours post delivery, you can see that the dominant part of EVs are co-registered with the tumor. So it is clearly indicating that the EVs are like going into the pathological region of the brain. In this case, is a GBM. And you can see all together in one image actually. There are 2 mechanisms by which anything delivered via nasal route will be going into the pathological region. The one of the pathway is called olfactory neural nerve pathway, and the other one is trigeminal nerve pathway. That is the one you have seen in our previous images. So the whichever is going directly into this one, directly into the tumor, they are via the olfactory nerve pathway. But whenever it is going to the back side, then it is trafficking towards the pathological region, that is via trigeminal pathway. So this is very clearly shown by this delivery approach. And we also tested like a kind of response rate by using MRI, since we have all different high resolution in vivo small animal MRI. we found that actually you can see many of the tumor in the complete treatment group, they kind of like reduced, even some cases almost eliminated. So we did ex vivo to confirm that actually those regions, is there any residual tumor? And this is the control complete treatment group in one animal. You can see that there is not at all a tumor. There is no even recurrent tumor. But in other cases, you can see this is a clear tumor location. And we also kind of quantify how much RNA delivered to those locations by using like a real-time PCR methods. And we see a huge amount of microRNA delivered to the tumor locations. So to support like a kind of our trigeminal and olfactory neuronal pathway of delivery to the tumor region, we did ex vivo histology of the tumor. So this is like the whole brain actually we take, and then we kind of soften the bone tissues with everything. Then we intact actually to stain for the ICG signal. And you can see here, this is the trigeminal pathway. And wherever you see red color, actually, that is the trafficking of the EVs, which is going towards the backside. And this is like a direct olfactory pathway. You can see these red colors, they are going to the directly to the brain region. And this you will see in this image much better actually. This is trigeminal pathway, this is olfactory pathway. So this is very clearly supporting that what we deliver via intranasal route is reaching this GBM region. And we also did a photoacoustic imaging to see the ICG signal in the brain tumor region. And you can see that control EV, the signal is much lower. But when you deliver by using the engineered EV, they are much, much higher. So this is like a movie which shows the photoacoustic imaging. And you can see because the photoacoustic imaging is a high resolution, so that you will able to see even very small location of the tumor. So I think I convinced that delivery of these EVs, microRNA delivered by EVs. Let me move on to the second application where we use like a kind of the second major type of vesicles. They are called apoptotic bodies. So as I said, actually depends on the condition, depends on the source cells these EVs are delivered. They tend to carry different type of cell surface or EV surface markers. So the EVs which are coming through these organized releasing mechanisms such as the microvesicles and exosomes carry different type of surface markers. But in the case of apoptotic bodies, they carry a lot of like annexin and other cell surface markers due to the apoptotic mechanism. Naturally, if our body there is any apoptotic cells or apoptotic bodies, they tend to attract macrophages for the clearance. So that one of the common problem which we normally experience is the drug resistance by the bacteria. So most of these bacteria, they develop drug resistance due to the inherent mechanism of modification to the bacterial cells. That include like a kind of active efflux of the drug, or modifying the cell surface for not allowing this drug to enter or antibiotic to enter inside, or they also do like a degradation of the antibiotics inside the cells by upregulating. But the other mechanism by which bacteria escape from the treatment of antibiotics are the hiding inside the macrophages or hiding inside other cells. One of the common hiding of these bacteria also happens in the cancer cells. So here you can see a streptococci, which is located inside like a kind of macrophages. So these green all are like a streptococci inside the macrophages. Similarly, we also see a lot of like a kind of TB, which are hiding within the granuloma in tissues. So this is one of the default mechanism. Many bacteria, they tend to hide within the cells so that antibiotics cannot enter the cells to kill them. So naturally, macrophages tend to like a kind of eat these apoptotic bodies as a natural clearance mechanism. So if you are able to load antibiotics inside the apoptotic bodies, you are able to like a kind of default activate these macrophages to take the antibiotic loaded apoptotic bodies. So indirectly, you are enriching the antibiotic load inside the macrophages. So the macrophages normally eat these apoptotic bodies through a particular signaling mechanism is called find me signaling mechanism. This is something like a kind of opposite to the one normally you see like a kind of in cancer cells. So that they eat these apoptotic bodies and they go inside the cells. Naturally, they will be like a kind of lysosomally degraded and released as a protein and nucleic acids. But in this particular case, which you load antibiotics inside these apoptotic bodies so that you are enriching more and more so that the intracellular bacteria can be easily killed. So we adopted our natural, the truck loading microfluidic based reconstruction mechanism, and we used like kind of streptococci aureus as our bacterial pathogen, and we used macrophage specific intracellular pathogens and cancer cell specific intracellular pathogens to treat. So we loaded vancomycin as an antibiotic using our reconstruction process. So if you have seen in the previous one actually where the extracellular vesicles are like a kind of bigger and smaller particles, when you see apoptotic bodies, their structure is completely different because their size is also very big. They can be like a kind of 1 to 5 micrometer in size. And if you compare between the previous image versus this, you can clearly see that the surface is completely displaying different type of architecture. So we induced these apoptotic bodies by using a starvation mechanism. So we isolated apoptotic bodies, and after starvation in different cancer cells, we tested how much of the apoptotic mechanism is activated. So we use normally FACS analysis. This is the control cells. When you use propodium iodide, apoptotic cells tend to take up more of this the fluorophore. This is like a kind of fluorescent dye. So it is indicating that when you do starvation, these cells are induced to apoptosis. So we isolate these apoptotic bodies from these induced apoptotic cells, and then we tested them whether they carry this annexin, one of the standard marker that apoptotic cells tend to have. This is like a kind of inner cell surface marker. Normally in normal cells, they are flipped into outside when the cells undergo apoptosis. So it clearly supports that actually these markers are overexpressed. So we use this to reconstruct and load. After reconstruction, you can see they are very uniform size and they tend to carry all different like a kind of structural features, everything similar to what we have seen with like a cancer cell EVs. And we also tested how much of this truck are loaded in these apoptotic bodies. So you can clearly see the trucks are loaded at very high quantity. And we used them to kill intracellular pathogens. First, we generated macrophages which are carrying this the Streptococcus aureus. And you can see here there are different conditions. And after treatment, you'll see that the one carry different cancer cell specific apoptotic bodies. They tend to kill more of these intracellular pathogens. We used a live dead cell assay. Wherever you see green, that intracellular pathogens, you can see that very dotted structure, they are bacteria. And wherever you see more red color, that is like a kind of dead cells, bacteria which are induced to death by these antibiotics. So we also tested in other cell type, actually cancer cell type. You can clearly see here, and this green all are bacteria, and whenever you treat with antibiotic you see a lot of bacteria which are dying inside the cells. You can see this third merged image, both live and dead cells, and you can see here a lot of cells are still undergoing apoptosis and cellular death mechanism. So now how whether you showed that actually intracellular pathogens ex vivo in a cultured condition. Now, how do you apply them in vivo? So now since they're like a kind of most of these bacteria related studies are restricted actually in animal house, we need a special approval. We showed that actually how they are biodistributed in vivo, whether they are taken up by the macrophages by intravenous injection. So after intravenous injection, you can see that they tend to accumulate in the liver. And when we tested them ex vivo, you can clearly see that spleen, liver, they tend to accumulate very high amount of these particles. Since spleen carry a lot of immune cells along with macrophages, they have very huge amount of these macrophages, apoptotic bodies loaded with vancomycin. And we also checked, actually, we used as a marker protein. We loaded microRNA along with these apoptotic bodies, so that we use this as like one of the mechanism to kind of test how much of those EVs or apoptotic bodies are delivered inside the cells. And we also intraperitoneally injected these apoptotic bodies loaded with vancomycin. And we also, after certain time, we isolated the peritoneal fluid, which carries a lot of macrophages. And we looked for how many of these macrophages they kind of engulf or eat these particular apoptotic bodies. You can see here, wherever you see this kind of colored structure, they are macrophages. They kind of they eat like a kind of this apoptotic bodies loaded with vancomycin. So it clearly supports like a kind of various assay systems. There are macrophages specifically kind of taking up these apoptotic bodies loaded with vancomycin. So to further confirm that, actually whether those apoptotic bodies are carrying a lot of different proteins related to the apoptotic mechanism, we did mass spec analysis. So we looked for different pathway regulated proteins. Any of these vesicles, if you take, actually they tend to carry a lot of different housekeeping proteins. Along with autophagy and apoptosis are a related mechanism, you see a lot of enriched proteins related to these pathways. So you can see that many of them are cell surface, some of them are cytoplasmic and extracellular space. So these are like a kind of distribution of these proteins within different locations of the cells. So finally, I just want to close a little bit with how we can use this for other applications like cancer therapy. So one of the common cancer vaccine or cancer vaccine immunotherapy is like a dendritic cell vaccine. So dendritic cells are like a kind of one of the antigen presenting cells. So dendritic cells are commonly used to treat cancer by treating with cancer specific antigens. So if you isolate mature dendritic cells and expose them to cancer cell specific vaccine or cancer cell specific antigen, they carry the receptor will carry these antigens. So that if you reintroduce these antigens, they go and kind of activate many of these cytotoxic T cells as well as the CD4 T helper cells. So they tend to produce a lot of anti-tumor immune response. So this is one of the common mechanism which is happening in naturally. When any patient receives chemotherapy or radiotherapy, they tend to like kill tumor cells. So when those tumor cells are killed, actually they tend to release antigens. So those antigens are again presented by these dendritic cells. So this is one of the kind of common technology used for treating patients with dendritic cell cancer vaccine. So this is like a kind of workflow normally adopted in the clinic where they tend to isolate monocytes or immature dendritic cells. They like a kind of treat with a cytokine cocktail to induce them to mature. Then they expose to tumor specific antigens and they reintroduce to the patient. This is a routine process used in the clinic. So, but the major problem with this particular approach is having enough number of these dendritic cells because they are very, very minimal number within our body. So that we adopted actually a strategy by using that actually we can isolate dendritic cells which are pre-treated with cancer-specific antigens, and those antigens can be introduced to a biomimetic microbubbles so that these microbubbles can carry or mimic like a dendritic cell vaccine. So now you are able to generate large number of microbubbles with a minimum number of dendritic cells. So by using this strategy, you will able to amplify the antigens, number of antigens by using this strategy. So we create this kind of microbubbles through a chemical conjugation and reconstruction process. By using this process, you have the flexibility of modifying this microbubble in several ways. You can have chemotherapeutic drugs, you can have microRNAs along with your tumor specific antigens. There are several ways you can modify these biomimetic microbubbles to improve the kind of therapeutic vaccine against cancer. So by using this strategy, we deliver. This naturally, these microbubbles are tend to accumulate more and more in the spleen, lungs, and the liver. So that once they delivered into the spleen, lungs, you can also use low intensity ultrasound with high pressure to break those microbubbles so that you are able to kind of increase the amount of dendritic cell specific microbubbles into the target immune organs. So we create these kind of microbubbles which are carrying dendrite cell antigens by using a targeted microfluidic process. In this process, we create these microbubbles, and we used like a kind of tumor-specific antigens carrying these microbubbles. So we adopted this strategy to treat in vivo. So to see that actually before and after display this dendritic cell vaccine, how that architecture of these microbubbles are changed. So you can see this is the control microbubbles, which are kind of, you cannot, because since microbubbles cannot be taken by using TEM images, we used SEM images because the high vacuum can break this microbubble. So if you look at the difference between this versus the one treated with dendritic cells, you can see here a lot of these small particles are the membrane carrying the antigen. So we used this strategy by isolating the dendritic cells, which are like a differentiated by using these cocktails. So we isolated from 3 different donor patients, and you can clearly see here without cocktail, this is immature dendritic cells, and this is after activation by using the cocktail. And they are exposed to tumor specific antigens, and we used them for reconstruction. So we used this for the respective tumor treatment. And you can clearly see that the one received immune microbubbles, they showed huge kind of reduction in the tumor volume. And they also improved the survival rate. And we reconfirmed this by ex vivo tumor size. You can see here, the one without any treatment, the tumors are very healthy. But wherever you see the immune microbubble, most of the tumors are showing much, much smaller size. We also ex vivo isolated the immune cells and tested how the system is activated. The normal one is that the immune activation is CD8 and CD83. So that you can see here, the one treated showed the high amount of immune cells, activated immune cells. So with that, actually, since I am running out of time, I want to like kind of summarize that by using this particular immune, like the cancer cell or the cell-derived vesicles for successful microRNA delivery into GBM for treatment. And also we showed that actually how we can use apoptotic bodies derived from cancer cells or other cells as a like antibiotic carrying molecules to kill the intracellular tumor. Or you can also adopt this for immunotherapy by using dendritic cell vaccine. We also work on like a kind of tumor cell derived engineered vesicles for various cancer therapy, but I'm not discussing here. With that, I think I will acknowledge, because all these things are not possible without support from many of my students as well as the collaborators. We work a lot with the UC Davis in adopting many of our technology in liver cancer and breast cancer in the DOC model, so that one of the model which is more similar to human. So if you are able to like kind of deliver sufficient amount of many of these therapeutic agents in dog tumor, then it is easily translatable to human. So we do with UC Davis. And we also want to acknowledge, I want to acknowledge a lot of different funding support from Focused Ultrasound Foundation, NIH and NCI, and one of the company we work with Japan actually to develop the recent CRISPR mediated gene knockout to improve cancer therapy. So with that, I thank everyone listening to the talk as well as for the invitation to present in this webinar. Thank you, everyone. I am ready for taking any question.
Thank you very much, Dr. Paul. Very comprehensive talk, and I learned a lot from this talk. So let me see if there are any questions in the chat. Otherwise, I don't see any. So I can start with a question. So since I am a clinical person, so my question to you is in the context of cancer immunotherapy.
Mhm.
How do you think the biometric nanovesicles avoid unintended immune suppression or tolerance when repeatedly administered?
Yeah, normally what happens actually, the cancers are developing through immune evasion. So they tend to express like a kind of tumor-specific antigen at the lower level during the development process. So over the time, actually, they educate our immune system in such a way that actually they are autologous antigen. So because of that, actually, our native immune cells or innate immune cells are not able to recognize them as like a kind of unusual antigen. So in this particular case, what we try to do is actually we select several different tumor-specific antigens and give them along with a specific customized adjuvant what we have developed in our lab, so that you are suddenly boosting your immune response against those antigens in a targeted way, so that you are able to like a kind of boost immune response very rapidly to those antigens. So they are tend to attack the or activate the immune system suddenly that was previously masked by the native kind of tumor specific antigens. So they tend to activate more and more. Did I answer your question?
Yes, thank you very much. Any questions from the audience? Because I don't want to keep on
Uh,
repeating. I
yeah,
have,
I'm Raghu Halkar, Saima.
yeah, Dr. Halkar, please go ahead. So,
Uh,
Dr.
uh, Thank you for a nice lecture and a comprehensive lecture. It gives us a whole idea of what's going on in the research field. Um, Currently, in nuclear medicine, the two popular therapies in the last 4 or 5 years in US has been Lutathera and Pluvicto, but they are targeted molecular therapies, and none of them use any nanotechnology. Because many times nuclear medicine molecules are much smaller than the nanoparticles. And
Yeah,
the only thing by what we suffer and what we need help from the nanotechnology is to minimize side effects. If the nano carriers, whether it's a bubble or it's a microsome, if it can somehow selectively go to the target and not go to other
yeah,
organs like salivary gland, that would be helpful. Are there any research going on in that direction
yeah.
to
That is the main idea actually, because nuclear medicine most of the time they use peptide or small proteins, since they want like a kind of rapid clearance and easy for FD approval and all those things. Because anything to do with this kind of nanomaterials, you need to justify a lot to the FDA clearance. So the reason why we want to adopt like a kind of this kind of natural cell derived vesicles, they are kind of almost similar to what we have in our body, so that they are not like a kind of anything new. And you can also like a kind of customize them, isolate from the same patient, and you can reintroduce them to the, and you can also engineer them in such a way that actually they display. So we engineer them like in this particular case for the GBM therapy, we used CXCR4 overexpression. Similar to that, actually, we have a lot of different antibody construction on the surface so that they can be specifically delivered to the target location without going into the non-specific accumulation in other tissues. So if you think about the natural cell membranes are nothing but actually they are phospholipids with integrated proteins, or in many cases they are like a kind of glycoproteins. So those phospholipids and the proteins are naturally kind of assembled through a kind of natural assembly process. So we exploit this particular process by using target proteins. They are conjugated to phospholipids, so that even after you functionalize with these antibodies or small targeting agent, they still maintain the natural integrity of the cell vesicles. So that I think we have a mechanism to selectively target the tumor locations or other pathological region by using this kind of engineering approach. Did I answer your question, Raghu?
Yeah, sure. Thank you. And probably the a combination of nanotechnology and targeted radionuclear therapy would decrease the side effects.
Yeah, that can reduce the side effects and improve the specificity.
Yeah, Dr. Halkar, there are several centers that are actually trying to harness nanotechnology with theranostics to improve targeting, especially in difficult to localize organs like the brain for METs as well as GBM. Unfortunately, it's still not yielding the results we want. So,
yeah, that is one of the reason actually. Anything to do with the brain, I think the blood-brain barrier is the major limiting factor. So that is the main reason actually we are trying to adopt intranasal delivery. So intranasal delivery, we are kind of giving like a 1/100th of the dose what we normally tend to inject systemically. So that, the toxicity is also reduced and it is more specific only to the brain actually. So, and also like in our body, I think lungs and the spleen and other lymph nodes are carrying large amount of immune cells. So anything if you deliver to these organs, they naturally straight away present the antigens to those immune cells. So rather than having like a kind of recruiting the site where anything to do with intramuscular injection vaccine, you need to recruit immune cells to the site of injection. So that process can be eliminated when you give anything via intranasal, either to the brain or to the lungs. So we also noticed actually, because when we give intranasal vaccine, which we did for our SARS-CoV-2, we developed and published in IASNM, that produced like a kind of 1,000 times stronger IgA response when compared to systemic or intramuscular injection. So the IgA is one of the strong, like a kind of neutralizing antibody that is more effective when it comes to the infectious diseases. So that even in cancer also, if you have more and more IgA, that anti-tumor response is much better. So that we have the data recently, we are working on the publication.
Interesting. I think Harshad has a question, Saima. He has a hand up.
Hey, hi.
Yeah.
Thank you. Thank you, Professor PAULMURUGAN. That was a great lecture. We really learned a lot. So, just to touch upon Dr. Halkar and Dr. Takalkar's points as well. So, have you? I mean, this field is obviously new for me as well. But how about the radio labeling of the nanovesicles to really ensure? Because you mentioned this for drug delivery as well. So, for targeted delivery of something like short-range alpha radiopharmaceuticals or Auger emitters directly to the tumor cells, and then is there the possibility of whole body imaging, to study the biodistribution using PET? Is that something which has been studied?
Yes, yeah, exactly. We are working on that actually, because we have the microfluidic based reconstruction process is very rapid. Actually, you can quickly reconstruct and either load your radionuclide, or you can also conjugate the radionuclide to the target protein and you can integrate with the vesicles. That we are working on to see that radio imaging of biodistribution as well as target specific accumulation. Yes, that we are working on. Yeah, thank you.
Thank you.
Yeah.
So as the field is growing, Dr. Paul, what do you envision? Like, what are the major bottlenecks that you are encountering, or what do you think would be the major kind of bottlenecks in the development of these nanovesicles?
Yeah, in the space of like a kind of cancer therapy. So you know that I think the one of the workflow developed in Europe actually, they normally they customize cancer vaccine through a kind of standard workflow. They take the tumor, they sequence the entire tumor cells, and they look for neoantigen and tumor-specific antigens. They synthesize those neoantigens and tumor-specific antigens, and they reintroduce back into the patient, the same patient, for the customized therapy or personalized therapy. So in that line, actually, we are working on looking for like a kind of neoantigens. You know, the difference between neoantigens versus tumor-specific antigens. Neoantigens are small peptides. So within a proteins, any protein with neoantigens, actually, they tend to have more specific for the tumor. They are not expressed in any other cells. So we are looking for a library of neoantigens and tumor-specific antigens that is more common in many cancer, so that we can make like a kind of on-the-shelf vaccine for many different cancer. Otherwise, that workflow which currently adopted in Europe, it's quite expensive. It is taking more than 6 months to come up with final therapeutics. So that is kind of too long for waiting a patient to get the treatment. So that is why we are working on that direction by integrating with our nanovesicles, so that it will be able to like a kind of on-the-shelf vaccine for different cancer patient.
Thank you.
I think you also mentioned microbubbles, but I think you were kind of developing them internally or something. But targeted microbubbles have been around for almost 3 decades, and they've been also tried as a drug delivery vehicle for various cancer as well as non-cancer diseases, including IBD or Crohn's disease and so on and so forth. But it's been 25-30 years, but we still don't see a FDA approved commercial product, and there's something that holds that technology back, and I'm suspecting that's a similar parallel for nanoparticles as well, because there have been nanoparticles around for some time as well. So there's something that still we are missing in harnessing that.
Yeah, these are all kind of the process with the FDA, okay? Because currently there is no targeted microbubble approved in US for application. So US only the non-targeted microbubbles are used for the perfusion imaging of liver, all those. In Europe, some of these VEGF targeted microbubbles are approved for clinical use, but in US actually they are not. Nothing is approved. It's a very slow process in US. And the major limitation in many of these targeted microbubbles, most of the targeted microbubbles are functionalized with antibodies. The surface antibodies, which are either targeting the vascular VEGFR or vascular CD31 vascular marker, which are overexpressed. So they have like a kind of long circulation time, even though the microbubbles are diffused after few minutes. But these antibodies are circulating for longer time, they tend to activate the immune system. That is the main reason actually our lab, in collaboration with one of my colleagues, Jeremy Dahl, we work on like a kind of developing small affibody as well as the nanobody based targeted microbubble for diagnostic imaging of breast cancer and liver cancer and pancreatic cancer. So we are working on that, because since the advantage with these small nanobodies, they clear very fast and they are very minimal in immune activation, non-specific immune activation. And anything to do with the microbubble, they are applicable only for imaging anything vascular target, because they are very larger size. So they don't like a kind of come out of the vasculature. So to adopt like a kind of tumor specific target will not work. That is the reason actually we also work on nanobubbles. We have published that paper in recently, one of the paper where we developed nanobubbles. They are very small, 100 nanometer size. Once they go out of the tumor, you can apply some ultrasound pressure to expand, and so that they tend to create contrast. So it is like a kind of activatable nanobubble. So by using this kind of strategy, the limitation as to answer your question, why it's not moving very fast, it's all the slow process with the FDA and approval process. So whenever you go from like a kind of a single molecule drug, if you take a small peptide, the approval process is much faster. When you have multiple proteins, so if you take the nanovesicles or if you take microbubbles, they involve a lot of different reagents. They need to be like a kind of studied very well before they are expanded or they are translated into the clinic. That is the main reason actually many of these nano platform, they are all with mixture of different chemicals. They are kind of slow in process.
Interesting.
Yeah.
Well, hopefully we'll see something down the road.
Yeah. We are expecting.
I have a slightly unrelated question. It's more about research technology and methodology. The West has always used a divisionistic approach for finding the answers. That is, take a big problem, subdivide into small, and answer those questions. Just like as you showed in your nice way, how from molecule to cellular uptake, and then go for an organ uptake, then do an animal study, and then do a human study. But many times, what works at a cellular level, it may not work at an organ level. It may not work at an animal level, because 10 cells together may not behave like 1 + 1, 10 cells. It may work totally differently. So are there any changes in the technological or research approach to more look into it a holistic way rather than a divisionistic way? Has there been a paradigm shift?
Yeah, I agree with you, because if you are able to like a kind of convert directly from anything, any results you get from the cells are able to translate into the clinic, means we have a lot of different therapeutics by now. So even I think small animals. Small animals, many times we cured the tumor, but they are not really going into the clinic. So the pharmacology and the bio kind of physiology, everything is completely different. So the holistic way, in the sense actually how we can translate into the clinic, or what is your exact question in the?
What I would aiming at, the way nuclear medicine has been able to bring certain things into a thing is by a theranostic way, that use something for diagnostic purposes and then apply the theranostic purpose. So that doesn't require too much of because in a diagnostic procedure, we are already getting a in vivo map of the biodistribution. So that's what I was aiming at.
Yeah. Many of these current
theranostic as a tool to develop new therapies was my reason behind the question.
Yeah. That's how we move actually, because so anything to do with the diagnostic way, your amount of the agent which you are injecting is in very trace amount. So the justification you need to provide is much lower. But when you want to translate that into therapeutics, so you are injecting like 1,000 times or sometimes several thousand times more than the amount we injected, right? So most of our approach starts from the diagnostic way, so that then we expand and see that how they are activating. That's how we work with microbubbles. Originally, we developed the diagnostic microbubbles. Now we are using them for the therapeutic. So that is the only way to do it actually. But the justification, FDA is having completely different view. When you ask for a diagnosis, it is easy for approval. But when you want to apply it for therapeutics, actually the approval process is very tedious.
Thank you.
Yeah.
All right. With that, thank you very much, Dr. Paul, for your talk. We all learned a lot from it, and thank you to all the audience and the people who took our time to be here. So with that, I'll say have a nice rest of your day, and we'll see you next time next month.
Yeah,
Thank
thank
you.
you so much. Thank you to everyone for attending, as well as thank you for the organizers. Yeah, I'll talk to you soon. Actually, I will see you in the next meeting.
Thank you, and Happy New Year to all.
Yeah, thank you.
Thank you, Dr. Paul.
Thank you.
Thank
Thank you.
you. Happy New Year all.
Yeah, Happy New Year to all. Bye.